It's great to be here, Dan. Thanks very much for having me.
Oh, you're very welcome. I've had you on my Fox show, my radio show. I had you on the Fox show a while ago, and you had discussed some of the homework and the cleanup job you were doing on some of the things that were happening over in the Wuhan lab. And one of the things you had noted was that in the Wuhan lab, there was some evidence that they may not have only been working on coronaviruses and the series of coronaviruses, but also more deadly viruses. One of them, notably called Nipah with an N, and that the fatality rate of this virus is anywhere from 50 to 80%. When you had said that on my Fox show, I got a boatload of emails from people who are understandably terrified. I mean, coronavirus has been awful, but it's certainly, thankfully, nowhere near 50 to 80% lethality. Should we be concerned about another super virus coming out of not just this lab, but other labs?
Well, I think we always should be. It's horrible to talk about considering yourself lucky with COVID, but this virus has about a 0.7 to 1% lethality rate now that we've settled into knowing about it. But you're absolutely right. In patient samples that were sent to the Wuhan Institute of Virology December 2019, they were sequenced on a machine that looks very deeply at not only what's in the patient sample, but sort of everything in the lab, because it picks up, a single dirt flake. So you can analyze 55 million reads from the machines that were uploaded into the database. And the bottom line is that the lab is widely contaminated with the Nipah virus, and it's not just the plain old infectious Nipah virus, but it's in cloning vectors. So the tools that biotechnologists use to move things around, to do gain-of-function research, to make things more airborne, et cetera, there's both the vector that you can buy from a catalog and do your genetics with, and the virus itself. So this is perhaps the most dangerous thing I have seen in a laboratory, because it was in the laboratories where patient samples were. It was by definition not BLSA4, the highest safety level. It was either 2 or 3. So you've got, a very deadly virus being experimented with, perhaps doing gain of function. All I know is they were moving the genes around in the way you would do with gain of function. I want to be careful about what I say. But it's, as you say, a 50 to 80% lethal virus.
I mean, this really is horrifying information. We have viruses out there right now. This is a respiratory virus, obviously coronavirus, but this is not the be-all, end-all in viruses. You have Hanta, you have Nipah, you have Ebola. I mean, these viruses transmit via different vectors. Obviously, you can have waterborne illness, insect-borne illness, but the fact that they're working on this stuff and there was evidence of some manipulation, you would think the United States government, and I hope they are, Doc, I hope they're just not telling us, has been really focused on this, but thank you for updating us on that. One of the other things we had discussed about previously, which I've been getting a lot of questions on, is you had said something in a radio interview you did with me about how coronavirus, it secretes a form of a protein, which does something to suppress either the immune or inflammatory response, and how you thought that was odd. It was a fascinating point. If you could just, now that we have some more time, we're not compressed by radio commercial timelines here. What did you mean by that? Why is it so unusual?
Okay, well, let's go back and learn a little bit about viruses and then get into it. There are 3 kinds of proteins in the coronavirus. 4 of them that end up in the baby viruses that go on to further infections. About a dozen that are inside the cell that are making the virus particles themselves. But there are 2 of them that are secreted by the virus into the bloodstream. They circulate in your bloodstream, and they're designed to do things to suppress the immune system. So it's very unusual for a virus to sort of put out this kind of false flag, as it were, almost from a protein point of view. When you look at the proteins the virus is making in the first 12 hours after an infection. So you got to assume the virus is doing the things it thinks most important in the particular time frame. The largest amount of protein it's making is this ORF8, ORF8, which is secreted in the bloodstream, and it does 2 things. It suppresses interferon response, and it suppresses the presentation of antigens to the immune system. So let's dissect those 2. When you get a viral infection, you get a fever and you get sweats and you feel like, you have that achy feeling. It's not the virus doing that. It's your own interferon. And it probably relates to a time when we didn't have medicines, of course. So this was a signal that you needed to go away from your tribe. You needed to go off in the woods and either live or die and then come back to your tribe. So it became a signal of infection. ORF8 is the only protein I've ever seen in a virus that suppresses this. The consequence is that SARS-CoV-2 is the first new respiratory virus that was asymptomatic from the beginning. Very unusual. What is the other thing it does? The other thing it does is it suppresses the presentation of antigens to the immune system. So when you get an infection, when you want your antibodies to be made, or you get a vaccine, you want your antibodies to be made, you want your T cells to respond, there's a presentation process. So there's a whole cell and a mechanism where this foreign protein is presented to the immune system. It says, immune system, this is something you need to make. You need to make antibodies against, you need to make T cells against, because it's foreign. This protein suppresses that. Now, we've seen that before. What virus does that the most? HIV. So what does HIV do? You can never get an antibody response. You can never get your immune system going with HIV because it's constantly being tamped down. So back to coronavirus. You get a vaccine, you're getting about 12% of the whole virus genome in the vaccine. You're only getting a piece of the spike protein. There's 3 other proteins in the virus that you're not getting antibodies to, and you're not getting anything with respect to ORF8. So probably a fundamental reason why infected patients have such a better immune response to vaccinated patients is they're actually making antibodies against ORF8. Final data point is, when you look at the human antibodies in an infected patient against the spike protein, the other membrane proteins in the virus, or ORF8, ORF8 is about an order of magnitude higher. So the true infection causes a very strong immune response with this very rare protein. Again, other data facts is ORF8 was being studied really only in one place, 3 years, from 2016 to 2019, and that was inside the Wuhan Institute of Virology. So they were doing paper after paper on what does ORF8 do, how do we make it stronger, how do we make it weaker. Even some of the U.S. scientists who were cutting edge, top of the team for the rest of coronavirus research, were not looking at ORF8 like the Wuhan Institute of Virology was.
Oh, wow. Well, I have 2 takeaways from that, and please correct me if I'm wrong. I think it would be not irresponsible to surmise that we know they were doing their homework on manipulation of viruses in the Wuhan lab. That's not a controversial point. That's accepted as fact. Whether it's conclusive that this was a lab leak, this specific virus, is not yet conclusive. A lot of evidence in that regard. But Doc, if I were doing experimentation with what I wanted to be a contagious respiratory virus, and I was creating some kind of a bioweapon, what I would do is I would make sure early on that patients weren't symptomatic and were in fact spreading it widely. I mean, what you're saying about the human immune response is correct. I'm not an immunologist, but I've been fascinated by science from early on. It's these mechanisms where you present as an illness that some of them are from your own body to make sure you're aware you're in fact ill. And you're right, a more of a sociological response so you don't infect the rest of the community as well. But what better way to spread a respiratory pathogen than to suppress that response, at least temporarily, to make sure you're as infectious as possible to as many people as possible? I mean, that's not a conspiracy theory. That's happened with this virus.
Absolutely. And so, an example is with SARS-1, which is a related virus, about 90% related. There were no asymptomatic patients whatsoever. Out of the 8,000, there was actually one, and it was so unusual they published it as a case report. One asymptomatic patient. Here we had about 40 to 50% of the patients are asymptomatic. And again, that's just never been seen before in a natural coronavirus.
That's just so bizarre. The second thing I take away from that is we're seeing a lot of data, and data matters. We're data-driven people, and we should be. But some of the data coming from foreign countries where the data sets are sadly more complete, we're seeing natural immunity seems to be not perfect, but more potent than some of the vaccine-derived immunity from either the mRNA or the whole virus J&J type vaccines, which are a little different, the technology there. Is that because of what you just said? A developed immunity to this separate protein that suppresses the response. Is that what you think that is, and why the natural immunity may long term be more effective than the vaccines we have now?
Well, yeah, Dan. I mean, I think it's the second layer of the bottom layer. The bottom layer of all immunology is that a natural infection gives you better immunity than a vaccine. I mean, vaccines are wonderful, and they've probably been next to clean water, one of the greatest public health successes of the last 200 years. But a natural infection will, by nature, be a better immune response. You'll get a broader range of B cells, broader range of T cells. The memory cells are a little bit stronger. It's like when you're in basic training and you have a war exercise, that's great. But if you get hardened by real battle, that's the difference.
That's a great analogy and an accurate one. Doctor, with natural immunity, something that concerns me, I had been vaccinated early on after a bout with lymphoma. I was one of the early vaccinations in Florida. I was one of the first past folks who got it. I had a pretty severe case. I don't know if other people have mild cases. I can't speak to that. I can only tell you my own personal anecdote. Mine was really bad. I mean, 103.5 fever, just chills, the whole panoply of awful things. I had the monoclonals. I also had a doctor who gave me ivermectin, hydroxychloroquine. His decision, my decision. I'm an adult. Well, you do you, folks. I'm not your doctor. I also took, vitamin C, vitamin D, zinc, quercetin, just over-the-counter things as well. Melatonin as well. Doc, I had it for basically 36 hours, and I was done with it. I didn't even miss work. I got it on Friday. I was back to work on Monday. But having said that, Saturday and Sunday were really awful. And what worried me is I started doing a little bit of homework on reinfection for personal reasons. I don't want to get reinfected with this thing. But It's not unheard of, sadly. Why do you think that is? I mean, just explain the immunological difference between, say, chicken pox, where, when I was a kid, they had chicken pox parties. Just get the kids now, get it done with while they're healthy. And you really, and you don't get them again. I know it's probably heresy to doctors, but I know that's what happened when we were younger. But why do you think the difference here where there are reinfections? They're not very common, but it does happen.
There are some mysteries with this particular virus. Part of it is that it's changing so quickly because we're both growing. We're doing 300 million probably gain of function experiments, right? Every person that gets infected, the virus plays with a particular that person's immune system, and then when it leaves that person for another, it's done a gain of function experiment. So it's rapidly evolving away from the immune system. It's dodging the antibodies that you're forming. So it's going to have to burn through its own repertoire. Now it has a relatively limited repertoire in terms of possible changes that can still bind to the receptor in a strong way, but get around the immune system. So I think Omicron, knock on wood, but it looks to me like it's going to be the last major wave before this truly becomes endemic, becomes something that we don't talk about, we don't hear about much at all in the news, simply because it's so infectious. But also so minimally lethal, minimally pathogenic. It's quite a good virus from that point of view.
Yeah, doctor, I mean, from your mouth to God's ears, I hope you're right. I mean, I really thought Delta down here in Florida was going to be the end of it. I mean, I have a friend who is a healthcare professional, works in one of the local hospitals, and, Delta was bad down here. And then I thought, gosh, they must be running out of people to infect. At some point, the simple arithmetic doesn't work. So I do hope you're right. That leads me to actually my next question, unintentional but perfect segue. Given Omicron, it appears, at least from the early data, we don't again have a conclusive data set yet, but that Omicron appears incredibly contagious, high or not here. But thank the Lord, less lethal. We haven't seen the degree of lethality and very serious side effects. Why is that? Why the upper respiratory rather than lower respiratory lung-based symptoms, which are more severe, that we saw with Delta and the original variant? What's the difference? It's the same core virus, granted some genetic differences. Why the disparity, thankfully, in fatality rate?
Yeah, so first let's talk about the infectiveness. So when Omicron came out, one of the remarkable things was that although it was missing a year worth of background mutations, as if it had been Rip Van Winkle or off in a lab somewhere, It had gained the ability to get around every single antibody that the general population had. It gets around all monoclonals except for one. This was a really unusual finding. It's the kind of thing that you would unusual in nature. It just doesn't happen in nature. Usually there's a short back and forth between the immune system. So as the virus drifts, the immune system drifts, and you sort of keep up with it. But in this case, it skipped a whole year worth of human experience. But in March of this last year, there was a paper from South Africa where they were purposely taking the virus from July 2020 and incubating it with convalescent serum, pooled serum from patients who had gotten over SARS-CoV-2 to see if the virus could still infect cells, even the presence of this pooled antibodies from the world's human population. Of course, that's a gain of function of getting around antibodies. So that's why it's so infectious.
But why do you think that the upper respiratory, is it an ACE receptor thing? Is it just targets different tissue types? Why not thankfully, the lower body, more serious inflammatory lung symptoms that have been literally suffocating people to death? I mean, people have been dying, not necessarily due to the virus, but due to the body's response to the virus.
Yep. So 3 observations, and then we'll make conclusions from those. Number 1 is it grows about 8 times faster in the upper airway and the nasal pharynx than the Delta variant. It grows about 1/4 as fast in the deep lungs as the Delta variant. And that's probably the reason it gives you an upper, a nasal discharge, sneezing, sore throat, but not the pneumonia. The other fact is that while it has the furin cleavage site that we've talked about so much, this thing that activates it, this thing that makes it go to the brain and the heart and the liver and all the additional organs that it goes to in addition to infecting a given human, the furin site on Omicron, which is kind of crazy, it's almost hard for me to say it, is not cleaved. So one of the other things that the cleavage of the furin site in Delta and the original virus did was this really, really vicious thing, which is called forming a syncytium. So when the spike protein is cleaved, the second half of it is like this battering ram that can go into cells, very, very easily. And when it's pre-cut, like it was in the original virus and in Delta, it goes into the neighboring cells. So when you look at an autopsy at a lung from a patient, what you'll see like 100 cells in one circle, because the virus has infected 100 cells right next to it, but it didn't do that by going out and coming back in through the surface. It went through the sides. This is called a syncytium virus. And this, of course, evades the immune system. This completely evades the immune system because it never sees the outside, but it requires the cleavage of the furin site. So Omicron cannot form a syncytium. All it can do is go out, deal with the immune system, and go into a cell in the traditional fashion. And this shows the fundamental difference of the cleavage of the furin site, both with respect to transmission, but also pathogenicity. You're not getting pneumonia, and you're not getting it, of course, into the brain and the heart and the lung and the vascular system, all because of the presence or absence of the cleavage of that 4 amino acid piece that was put in the Wuhan Institute of Virology.
Doc, you've been very generous with your time. Just a couple more questions for you. The trajectory of this virus going forward. Listen, I know this is all predictive, but you're a doctor. That's what we do. I mean, you don't know where cancer is going to go either. It doesn't stop you from treating it. We've seen with respiratory viruses in the past, notably the 1918 Spanish flu, which was particularly dangerous, that the people who were infected with that later on eventually burned itself out. We still have influenza, but we didn't see a virus as severe. The reason's pretty clearly an evolutionary one. You kill all your hosts, there's nothing left to infect. I mean, viruses don't think. Obviously, they're not alive. It's a DNA-coded protein, and things. It doesn't think. But isn't it more effective for this virus moving forward to become like Omicron, highly contagious and more contagious, but yet less lethal? And do you know of any examples in the research you've seen of viruses like this that have become the opposite, say, maintaining their infection level but becoming more lethal? Because that would be scary. That's what concerns me.
Well, I think Delta was approaching that kind of event, but Omicron is clearly very, very different. I mean, I think that I want to get some good news, out for your audience there. So there's a nice study
Please.
out of the Kaiser system in California. 52,000 Omicron patients, 18,000 Delta patients from November, 1st November, 1st of January. So that's a nice population. There was a 75% reduction in hospitalizations with Omicron versus Delta. Misspoke, a 50% hospitalization, 75% ICU, and a 91% death reduction. One person out of 53,000 died of Omicron in this study. And so that's the thing I use with patients is I try to put something in context they can understand. Your lifetime risk of being struck by lightning, 1 in 15,000. So your risk of being killed by Omicron, 1 in 52,000. I think we can go back to normal life with that kind of risk probability.
Yeah. Well, I concur, and I think a lot of people. You've seen, regardless, Doc. I know this. You're a scientist. You're not a politician. I don't ask you political questions, but thankfully, we've seen some people on both sides of the massive political divide say the same. I mean, even Gayle King was on a nightly show this week saying, "Listen, I'm done with this. It's time to just go back, take your mitigation measures, wash your hands, eat right, lose some weight if you need to." The old common sense advice doctors like yourself been giving people since, I saw my pediatrician, Dr. Suska, God rest his soul, 40 years ago. Eat your apples, Daniel, drink a lot of water. I mean, common sense stuff. Last question, Doc. I get a lot of emails on this. Obviously, me not being a doctor or medical professional. People are very concerned about some of the reports of myocarditis. They are rare, thankfully, but they are there. They're not non-existent. And the question I get a lot is, listen, I'm a young man, young woman. I can't answer them because I'm not their doctor. But is this worth it for me? And a lot of them have already had coronavirus. In other words, is it worth the risk? And I'm not asking you to get. I'm just asking you to comment on the general trend for some of these folks to get myocarditis. They are concerned. Some of them had corona. They're 18, 19. They're athletic. They're young. They're like, hey, you know what? Is this for me or is it not? What do you make of a lot of these reports about this inflammatory heart condition?
Well, it's real, and it occurs with both the virus and the vaccines. It occurs typically in young men more than women, about 3 to 1 men, male to female. It's typically mild. You typically recover from it completely, but if the heart cells can't reproduce, so to the extent you lose 1%, 2% of your cardiac output, it's gone. If you're an athlete, that's a really big deal. So if you've had, if you've had the infection, I think you're fully immunized. I mean, I am not. I've looked at the data on the boosters. I mean, if you want to get one, they're fine, especially if you're over 60, probably. But I think we've gotten as much as we can out of the original vaccines because Omicron has now moved away. So we know scientifically that the spike protein that the vaccines make is no longer recognized. The antibodies you make against that vaccine are no longer effective against Omicron. So I really think that at this point in time we should stop. Now, in terms of what you do, I think there's also this strange increase in all-cause mortality now
Right.
in many insurance companies where they have to write a check when somebody cashes in their life insurance. So they're Johnny on the spot on this particular topic. There seems to be, in my opinion, and this is now I'm going a little off the reservation, but that there's sort of a, how can I say it, an increase in the coagulation state. So if you were to measure the coagulation parameters in any given patient, it's going to be the same. You do 100,000 patients, there might be a tiny difference on a population basis. But what this means is that the second and third standard deviations are going to have more coagulation events. So what does that mean? That means stroke, that means heart attacks, that means any sort of coagulation, renal failures, and those sorts of things. There's actually been studies where very, very good hydration in post-stroke patients reduces repeat strokes by 15, 20, 30%. So I'm telling everyone I know, look at it, let's stay really well hydrated for the next 6-9 months. I'm not good at that. I don't like to I don't like to drink water a lot. It's just not my nature. But
Yeah,
I
me
think
either.
probably right now we should keep our blood as viscous as we can. I think a lot of people sometimes take a Bayer aspirin or a St. Johnson's aspirin at the bedtime. Again, to kind of make your blood a little bit slick. Our blood is thickest between 2 and 5 in the morning, and that's why you get strokes and heart attacks in that time frame. So common sense things, but I think there's a coagulation change that may be fundamental now in the population, completely manageable, but good to know about, in my opinion.
Yeah, over the last year, I've really focused on hydration too, and I think we'll end there, Doc, because it just, is this kind of where we came in? Reminds me of again when I was a kid, how the standard medical advice healthcare professionals for decades have been giving you still applies. Eat a balanced diet, work out. Don't let this coronavirus attack, fat tissues. Work out, try to lose some weight, hydrate yourself. These are all old standard tips that never, ever get old. Doctor, you are just a well of fantastic information. I have tremendous respect for you. You've been an asset to this show and our audience. Thank you so much for spending some time with us. You are welcome back anytime. Thanks a lot.